This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Mass selection, also called phenotypic selection, is the oldest plant breeding technique dating from the very beginning of the agriculture about 12,000 years ago. Thanks to this technique, farmers have been able to make spectacular transformation of some wild plants into today's cultivated crops. One of the most staggering examples might be the domestication of corn from its wild ancestor teosinte. Compared to the small stalks of teosinte that have only a few hard-to-digest seeds, today's corn has not only tender seeds but also a huge amount of them per stalk, and exhibits a fascinating variety of seed and cob sizes, shapes, and colors. Mass selection is a seemingly simple procedure. It can be applied to any crop population that shows heritable phenotypic variability in the field, to which an artificial selective pressure is applied by the farmer or the breeder. The selective pressure can be negative, so that undesired phenotypes are eliminated, or positive, so that only the most desired phenotypes are selected. The plants remaining or being chosen by selection are then left to reproduce for the following harvest. The selection process is repeated several years until the population exhibits uniformly the desired trait or a range of values that are desired. There are, however, two essential conditions that need to be fulfilled for mass selection to work. First, the population must have sufficient phenotypic variation in the field. Without variation, there is nothing to select for or against. But phenotypic variation on its own is not sufficient. This variation needs to be heritable so that the selection is effective. If phenotypic variation is not heritable, for instance, if it is caused by variations in the quality of the environment, then the selection will be without any effect, and the mean phenotype of the population will not change after selection. You can learn more about heritability and other quantitative genetic concepts useful for breeders in the playlist of quantitative genetics. Breeders also need to account for the effect of the environment on the expression of the phenotype when doing mass selection. A set of identical genotypes can express dramatically different phenotypes between two contrasting environments. Therefore, selection in one environment can result in a crop variety that is highly adapted to that specific environment, but not to a different one. Ideally, crops should maintain high performance and resistance across a wide range of environmental variation. To achieve this, selection can be done alternatively in different environments. For instance, the crop can be rotated among different environmental conditions every growth season. Another way to do this, especially for crops that are to be commercialized on a worldwide scale, is to grow the crop in a different location every growth season, each location being characterized by its own specific climate, natural pathogens, and other biotic and abiotic challenges for the crop. This kind of traveling breeding program can also help to reduce the time required to produce a new crop variety and is known as shuttle breeding. In addition to applying mass selection in different environments, and thus making a crop that can be highly performant across a wide variation of conditions, shuttle breeding is usually alternating growth at different hemispheres of the planet or at different altitudes. So instead of waiting a whole year until the new growth season arrives, the same crop can be grown twice in the same calendar year, taking advantage of the different timing of the growth season in each locality. The pioneering shuttle breeding program was done on wheat in Mexico, which was grown alternatively between the lowlands of Obregon late in the growth season and the highlands of Toluca early in the season. In addition to decreasing the length of the breeding program by half, because each year there were 2 harvests of wheat, the new wheat variety was insensitive to photoperiod variation and resistant to a wide array of diseases. The result of mass selection is usually a genetically diverse population with traits that have been improved in comparison to its ancestral population. Potential uses of mass selection are therefore to produce or improve existing crop plant races. A landrace is a variety of a domesticated plant or animal that has a set of unique, recognizable phenotypic characteristics but remains dynamic. This means that some variation is observed between individuals and also from one generation to the next. This is because a landrace is not genetically uniform. A landrace is usually tied to a local environment, hence the term land in landrace, and is the product of long-term mass selection of the species in that environment. So the landrace is locally adapted. It will show high trait consistency and overall good performance in its environment of origin, but might not be as productive or as resistant if suddenly transferred to a different environment. However, because of their genetic variability, landraces can easily adapt if moved to a new environment via the process of mass selection. Some landraces are genetically more homogeneous than others, meaning they show higher phenotypic consistencies. Under some conditions, if they fulfill the requirements by local agricultural and breeding authorities, certain landraces can be registered as cultivars. Cultivars show enough phenotypic stability, or low genetic variability, so that a certain level of yield performance is guaranteed, and therefore can be certified and commercialized for a broad use. Because modern legislation requires that crops used for human consumptions have stable, predictable characters, landrace crops are often underlying some restriction about their trade. Mass selection is not used for industrial or commercial production of human food. It is, however, used to produce forage crops that are used as animal feed. An example of this is the Marshall ryegrass that resulted from the mass selection of a small ryegrass in Mississippi, USA over the course of 29 years, during which each year the seeds of the biggest, most resistant stalks were used for planting the next year. Mass selection is also frequently used for breeding ornamental plants, whether it is willingly or unknowingly among amateurs. Intentional horticulture breeding programs have resulted in a huge diversity of floral shape, color, and scents. Amateur breeders could also, to some extent, practice a form of mass selection by sharing seeds or clones of the plants with the most interesting or original characteristics among breeders. Finally, mass selection can also be paired with other breeding techniques complementing them. Specifically, in the case of controlled crosses that are the first step to produce new, true breeding lines, mass selection can be used to eliminate undesired traits inherited by each of the parents. This will be discussed in the following video.